viewers. Today in this session, I'll be talking about the higher order structures of nucleic acids. I'll be talking about the tertiary as well as the quaternary structure of nucleic acids, meaning both DNA and RNA. Till now we have seen that DNA, which is a polymer of nucleotides, as well as RNA, which is also a polymer of nucleotides, can exhibit different levels of organization. Starting from its primary to secondary, now we will be dealing with the tertiary structures. Now, like any other biological macromolecule, as we can see here, the tertiary structure of a protein. So, similarly, the nucleic acids also exhibits their tertiary structure. These tertiary structure are nothing but the higher order folding of elements of regular secondary structures. Now, these kind of tertiary structures are very important in performing the normal physiological functions of an RNA or a DNA molecule. Let us see now that what do we mean by the supercoiled or the tertiary structure of DNA. When we see this kind of a feature or a diagram very commonly in the textbooks. That is, we find that the bacterial cell contains different types of circular, highly twisted chromosome. So, thus the genetic analysis of bacteria and viruses have shown that these kind of tertiary structures are present within the prokaryotic as well as the eukaryotic cell. Apart from this, the plasmid DNA, which are the extrachromosomal self-replicating DNA molecule, are also circular in nature and are highly twisted. Thus, these represent the tertiary structure of DNA. This is known as the DNA supercoiling. Thus, DNA supercoiling is nothing but the tertiary structure. Now, let us see that how does it takes place. DNA supercoiling generally takes place when a covalently closed circular DNA molecule folds upon itself to form a highly coiled structure. This is known as supercoiled DNA. Now, this supercoiled DNA can also be formed apart from the covalently closed circular DNA molecule. Now, suppose a DNA molecule whose ends are fixed, either by the formation of covalent linkage or its ability to bind to a protein. As a result, the DNA, since it is bound to the protein, now distort the DNA helix. Then what is happening? Due to the distortional stress or due to the torsional stress rather, the DNA helix becomes supercoiled. As a result, what happens when it becomes supercoiled? So, it results in the formation of twisted appearance. Now, it's a common thing. We find in this diagram, the lower panel of the diagram, the left hand side diagram shows that a relaxed circle can lie flat on a plane surface. But when the DNA becomes supercoiled in nature, due to its highly twisted, it cannot no longer lie flat on a plane surface. This is supercoiled DNA. Now, let us see that what are the different types. As seen in this slide, we can see that the supercoiling can be done in two ways, positive supercoiling and negative supercoiling. Positive supercoiling takes place when the DNA is wound in the right-handed direction, giving rise to overwinding of DNA. On the other hand, negative supercoiling takes place when the DNA is wound in the left-hand direction, giving rise to the unwounding of DNA. So, as a result, here the DNA is becoming tightly packed and here the DNA is becoming less tightly packed respectively. Now, in most of the native DNA, it is present in the form of negative supercoil as it facilitates the different kinds of physiological functions within the cell. Now, this picture we often find. Now, this is what this picture shows, that the two DNA strands are intertwined with each other and the ends are closed with the help of a covalent linkage. The number of turns that the one strand is passing over the another is fixed and is constant for a particular supercoiled DNA. This kind of a feature was first expressed mathematically by James White in the year 1969. He found that or expressed it as the geometric component or the sum of two geometric components, that is twist or wreath. So we can say that the linking number is nothing but twist plus wreath. What is linking number? Linking number is nothing but the number of twists that one helix makes up on another helix, or the number of turns that one helix makes over the or one polynucleotide strand makes over the another polynucleotide strand is known as the linking number. It is a constant topological property of the supercoiled DNA. Apart from this, the twist is defined as the number of revolutions that one polynucleotide chain is making about the duplex axis in the particular conformation being studied. Apart from this, when a covalently closed circular DNA molecule is torsionally stressed, it results in the formation of wreath. The wreath formation takes place because the long axis of the DNA molecule becomes twisted on itself in three-dimensional structure, thereby forming wreath. Now, twist and wreath are interconvertible. If we look at this diagram, we find that when wreath increases, twist decreases. When twist increases, wreath decreases. Thus, twist and wreath are interconvertible, keeping the linking number constant. The linking number can only be changed by breaking one or both the phosphodiester backbone of the DNA double helix. Now, the types of supercoiling. Apart from positive and negative supercoiling, DNA can be supercoiled in two major forms. One is the toroidal supercoiling, another is the interwound supercoiling. Toroidal supercoiling takes place when DNA is wound around a cylinder, whereas the interwound supercoiling takes place when the DNA is twisted upon itself. This is known as the interwound supercoiling. Now, toroidal supercoiling is very common in case of eukaryotes. We find it where? Mostly in the form of chromatin. In chromatin, we see this kind of a structure which is known as nucleosome. In nucleosome, the DNA is wrapped around the histone octamer, and the DNA which is wrapped around the histone octamer is left-handed in direction, thus thereby creating a toroidal supercoiling. This toroidal supercoiling serves as the center of accumulation of DNA-protein interaction. It serves as the center of accumulation of DNA in the form of ribbons, or maybe the DNA supercoiling takes place in the form of DNA ribbons which are arranged in the form of spools. Thus, they also play a very important role in regulating gene expression and in forming the chromosome structure from chromatin. Thus, the different types of supercoiling are present in different types of cells. Topoisomerase. Now we have, as I have already told you, that the linking number is constant and it is fixed for a given topological molecule, or the linking number is fixed for a given supercoiled DNA. Now the linking number can only be changed with the enzymes which are known as topoisomerases. Topoisomerases, as the name suggests, they cut one or both the polynucleotide strands and thereby they change the linking number. This is very clear of if we study this diagram, we can see the action of topoisomerase very clearly. We find that on the left hand side, when the DNA molecule is covalently closed, then the linking number is 36. But in the presence of topoisomerase, which converts into a supercoiled DNA, the linking number changes from 36 to 32. Thus, the linking number is changed. And this change is made possible only by the enzymes which are known as topoisomerase. Topoisomerases play a very important role in cell. They are utilized for every physiological function like DNA repair, DNA replication, DNA transcription. Everywhere the presence of topoisomerases is required. Now there are two types of topoisomerase, type 1, type 2. The type 1 is They make a single nick in the phosphodiester backbone, pass the other strand through the nick and then reseals it. Whereas type 2 cuts both the DNA strands, passes a portion of the DNA duplex through the cut strand and then again reseals it. Now, the measurement of supercoiling can be done by various means. One is through ethidium bromide. Ethidium bromide, whose structure is shown in this slide, contains 3 ring-like structure. The presence of this 3 ring-like structures allows it to intercalate between the coplanar purine-pyrimidine bases. It's been found that ethidium bromide at the site of insertion causes unwinding of DNA. Another important feature is that the different topoisomers can be separated through the DNA sedimentation technique. That is, whenever there is an unwinding, the twist decreases and the wreath increases. So, such a kind of a feature we find in this diagram also. Since here it is written ethidium bromide concentration increases, as a result we see that the supercoiling or the nature of the supercoiling becomes relaxed here. Again it increases. And the different rate or different types of topoisomers also affect the sedimentation velocity. So, in this way the different topoisomers can be separated through centrifugation. Again, what happens if we run or try this topoisomers or run this topoisomers on an agarose gel? We find that the DNA which is highly supercoiled migrate faster and the DNA which is covalently closed migrates less. Thus, the topoisomers which are chemically identical species or DNA, they are similar to each other since they have the same number of bases, but they vary when they are migrated on an agarose gel. Topoisomers produce a series of discrete bands and this bands allows it to distinguish. Now, this topoisomerases, as I have already told you, it's very important for the cell. There are certain compounds like doxorubicin, etoposide, they inhibit the topoisomerase. Now, since they inhibit the topoisomerases, they can be used as potent anti-cancer agents. Thus, this different kind of compounds are regularly used for the treatment of cancerous cells. Let us now see the quaternary structure. The quaternary structure of DNA represents another higher level order or structure of DNA molecule. This represents the interaction which is taking place between DNA and protein during the formation of chromosome. That is how the nucleosome model or how the nucleosome is getting folded to chromosome. So, this represents the quaternary structure of DNA. Now, let us talk something about RNA. RNA, which is also a polymer of nucleotides, generally contains different kind of secondary and tertiary structure. The important tertiary structural motifs are the tetraloop, the pseudoknot, bulge loop and so on. We will be discussing some of them. The first amongst them is pseudoknot. Pseudoknot is a complex tertiary structure. The pseudoknots are formed when two adjacent stem loop-like structures are formed. Now, in this, one half of the stem loop intercalates between the two cut halves of another stem loop, thus resulting in a complex formation, or when hydrogen bonding takes place between sequences which are non-contiguous to each other. As a result, it results in the formation of pseudoknots. Further on, this is a very important feature of RNA, which are known as the U-turn. U-turn helps in bending the direction of the RNA backbone. The U-turn motifs are found when RNA contains particular sequences like the UNR sequence or GNRA tetraloops. Now, let us see this diagram, UNR sequence. What is happening? We can see a U-turn is made between first and second nucleotide. This whole structure is stabilized by means of two hydrogen bonds. The first between the uridine iminohydrogen group with that of the phosphate group of the fourth nucleotide and following UNR sequence. The second hydrogen bond is formed between the 2' hydroxyl group of ribose sugar and N7 of purine. Thus, U-turn is stabilized by means of hydrogen bond. On the other hand, tetra loops are 4-base hairpin loops-like structures which are formed at the apices of double helices. Many kinds of structures give rise to tetra loops. The most common is GNRA. Now, this GNRA is the building block of RNA structure, and this is again stabilized by means of 4 hydrogen bonds. The first existing between guanine and adenine, the second existing between the guano amino hydrogen with that of the oxygen of phosphate of adenine. The third is between the 2' hydroxyl group of guanine with N7 of R, and the fourth one is the stacking interaction which is taking place between N, R and A sequence. So, thus with the help of these two particular base sequences, U-turn motif is stabilized. The U-turn motif plays an important role in giving a bend to the RNA backbone, in changing the direction of the RNA backbone, as well as it provides a scaffold for providing the tertiary structure or for promotion of the tertiary structure of RNA. Now, such kind of structures are very important. If we closely examine the functions of U-turns, we find that such kind of U-turns are found in T-loop, in anticodon loop of tRNA, in the hammerhead of ribozyme and so on. Now, as we can see, the nucleotides which are present on the 5' side of the T-loop base pairs with the bases present in the D-loop. Similarly, the nucleotides which are present on the 3' side of the anticodon base pairs with the bases present in the mRNA. As a result, both of these structures are stabilizing the interaction. Here we can see how U-turn is important in performing the physiological function of tRNA. The similar kind of a structure is also found in hammerhead ribozyme. Let us now see the tertiary structure of tRNA. The tertiary structure we have already discussed about the clover leaf model of tRNA. The tertiary structure is formed by the bases which are present in the D-loop, that is the bases of the D-loop when hydrogen bonds with the bases in the T-C loop, or the bases in the D-loop when it hydrogen bonds with the bases present in the variable loop. As a result of their coming close to each other, we can see the cloverleaf model gets folded to an L-shaped structure. In this L-shaped structure, we find that the amino acid stem is present at one end of the structure, followed by the anticodon stem which is present at the opposite end. The D-loop and the D-loop are found at the corners. Thus, the tertiary structure of tRNA is formed due to the interaction of the different bases which are present in the different stem loops of tRNA. Let us now see the quaternary structure of RNA. The quaternary structure of RNA takes place, or the best example of the quaternary structure is the interaction which is taking place between the 23S rRNA present in one ribosomal subunit with 16S rRNA present in another ribosomal subunit. The two subunits come closer to each other, and hence there is a quaternary interaction between the rRNA. Similarly, the rRNA along with the proteins present in the ribosome interacts with each other. This is again another best example of quaternary structure of RNA. Thus, this helps in performing the physiological function better. So, what we have learned? That is, the tertiary structure of nucleic acids plays an important physiological role in the cell. The tertiary structure of DNA represents DNA supercoiling. The supercoiling is found in two different forms. One is the toroidal, another is the interwound, found in different kinds of cell. The supercoiling has a fixed number, or got a fixed linking number which can be changed with the help of certain enzymes known as topoisomerases. Topoisomerases are very important for performing the cellular function, and hence the agents or the compounds which are acting against the topoisomerases can be used as chemotherapeutic agents. The quaternary structure of DNA represents the chromatin and how it folds to form chromosome. Then we have RNA. The important structural or the tertiary motifs of RNA is pseudoknots, U-turns, GNRA tetraloops and so on. All of these sequences are stabilized by means of hydrogen bond. The tertiary structure of tRNA is very important and its formation is required for the efficient functioning of tRNA. The quaternary structure of RNA involves the interaction between the rRNA subunits as well as between the different RNA and proteins, and thus it helps to perform the function better. Thus, with this session, we end the chapter on nucleic acid, and we see that nucleic acid, like any other biological macromolecule, is dependent on its structure formation. Whenever there is a proper structure formation, it is able to carry out its function more efficiently. Thank you.